Remember when I first introduced the immune system in episode 16? If, by some miracle, you do remember, then you get a gold star. If not, no worries. I got the perfect prescription, a fun deep dive into immunology, the area of science that studies the immune system, and our efforts to use vaults as vaccines. Immunology includes a vast network of organs, tissues, cells, and molecules that function together in intricate ways. Most research in immunology focuses on cells, receptors, and proteins involved in fighting pathogens. Immunology depends on cell biology to understand how immune cells work. When I was getting my training in biochemistry and cell biology, the field of immunology was so new that scientists working in this area were in the process of discovering, characterizing, and naming this amazingly complex system. These scientists created new terminology that served as a shorthand for describing the complexity of the immune system. A newcomer could be overwhelmed by the jargon. So, the CDX effectors express IL-13, IL-4, CCL7, and GM-CSF. Wow! The CDYs rely on CCL15 and IL-25 for their stimulation activity. Hi, I'm Lenny Rome, the Vault Guy. I'm a cell biologist and a nanotechnologist. My passion is a common particle of the human cell called a vault. With the help of my daughter-in-law, Tara, I'm trying to explain what vaults are to non-scientists. As a bonus, if you're following along with my lessons, you're also learning some basic cell biology, and with this lesson, some basic immunology too. Today, I want to cover three things. First, I want to discuss some important properties of vaccines that are necessary to understand in case you want to make a new vaccine. Second, I'll teach you about vaccine antigens and adjuvants. Finally, I'll describe the immune response elicited by a vaccine. Stick around, I'll tell you about our attempts to make anti-vault antibodies. You might be surprised by our results. Vaccines are very important as they can prevent contraction of an infectious disease. According to the World Health Organization, vaccination saves 5 lives every minute. There are several important properties that a vaccine must meet in order for it to be useful and successful. Everyone is aware of some of these properties, like they must be safe and they must be effective. But there are other requirements for vaccines that not everyone thinks about all the time. For example, the arrival of a new pathogen that causes an infectious disease often requires a rapid response. And therefore, the speed at which a new vaccine can be developed and shown to be safe and effective turns out to be incredibly important. The stability of a vaccine is also very important. Stability is a broad requirement covering vaccine potency and the ability of a vaccine to withstand storage and variations in temperature. Vaccines need to be shipped all over the world, and if a vaccine needs to be kept very cold for it to be stable, this requirement can seriously limit its usefulness. A vaccine that needs to be stored frozen isn't going to be useful in an African village that doesn't have electricity, let alone -80-degree freezers. Another problem can be the cost of large-scale production of the millions of doses that are often required for pandemic emergencies, and the time and cost to gain the approval for their use. A lot has been said about the mRNA COVID vaccine and how well or not this technology satisfies the requirements that I just talked about. Certainly, the speed at which the COVID vaccine was developed is a testament to the strength of this technology. When vaccines were first introduced, the technology was based on vaccination with a weakened or inactivated pathogen that could still trigger a robust immune response without causing safety concerns or the induction of a disease. We certainly didn't want to give anyone a mild case of polio to prevent them from getting a serious case of polio. Driven by the desire to lower the side effects of these vaccines, the technology moved to subunit vaccines, which involve using just a piece of the pathogen, most often a protein or peptide derived from the pathogen. This chunk of the pathogen that is presented to the immune system is called an antigen. Subunit vaccines were not very effective at getting a robust immune response, so they needed to be delivered with an additional component mixed in, called an adjuvant. The term adjuvant comes from the Latin adjuvare, which means to help or aid. Adjuvants were first discovered in 1920 at the Pasteur Institute by a veterinarian named Gaston Ramon. Ramon, who in this photo looks like a Pasteur Institute professor right out of central casting, observed that an inflammation, which is redness or swelling, at the vaccination injection site improved the immune response. A little later, it was observed that certain aluminum salts called alum worked well as adjuvants. We used to think that adjuvants acted simply by causing an inflammation that attracted cells of the immune system to the site of vaccination. Studies now indicate that adjuvants likely act by a combination of different mechanisms that enhance the recruitment of immune cells, the uptake of antigens, and their presentation to the immune system. Even though adjuvants are used in most vaccines today, their precise mechanism for boosting the immune system is still a topic of ongoing research. There's a lot of public concern that adjuvants may be the cause of some vaccine-related side effects. Our immune system responds in two different ways to vaccination. One is the production of antibodies. Antibodies, which I have described previously, are circulating defense molecules that can attack the pathogen. One nice thing about antibodies is they can stick around for a long time themselves, or we can develop the capacity to rapidly make that particular antibody in the future. This capacity is one example of the second type of immune response to vaccination. It's called cellular immunity. With cellular immunity, the vaccine triggers the production of various types of immune cells that can directly attack the pathogen or retain the ability to respond to the pathogen at a later time. For example, a cell called a memory T cell is remarkably long-lived and can remember a pathogen even decades after it was first encountered. As I continue to describe the potential of vaults as vaccines, I'm going to return to these desired properties of a vaccine. So, what does a vault vaccine look like and how are they produced? Well, none of this will be new to my avid vaulters and subscribers of my lessons channel. This is the technology I've been talking about since episode 14 and again in episode 19. Remember the vault zip code? There's another gold star for you. If you don't want to go back and review, I'll describe a piece of a protein that we call the INT or INT domain that we can link to any protein or peptide of interest. The INT-linked protein or peptide can be easily packaged inside the vault particle. For today's lesson, I'm going to refer to the foreign protein or peptide derived from a pathogen as an antigen. Just to be clear, these antigens are composed of amino acids. They are not RNA. We co-express an INT-antigen fusion protein and an empty human vault particle in the yeast system that I described in episode 22. The vaults that we purify from these yeast are composed of INT and MVP, two human vault proteins, and a foreign antigen of choice. Let's back up one step and ask what happens if we use native vaults as antigens. This was something that Nancy Kadersha and I did soon after we first discovered vaults. Nancy immunized rabbits with native rat vaults, expecting to see an antibody response, but she saw nothing, no immune reaction at all. We also looked for an anti-vault antibody in human patients by screening blood samples from a bank of human autoimmune diseases. Allow me to unpack that for you. Disorders of the immune system like lupus, rheumatoid arthritis, and type 1 diabetes are characterized by overactivity of the immune system. In these disorders, the body can attack and damage its own tissues. When we screened hundreds of patient samples from these types of disorders for possible self-antibodies directed toward vaults, we could not find any. Later, when we learned how to make empty human vaults, we also tried immunizing a rabbit with them, and we found no evidence that the human vaults caused an immune reaction in the rabbit. This was very interesting because rabbits and humans diverged about 100 million years ago. Over that 100 million years, human proteins have developed so many amino acid changes that the vast majority of them are recognized as foreign by the rabbit immune system. However, vault proteins are mostly unchanged even after millions of years, so the rabbit immune system doesn't recognize a human vault as a foreign object. All these results led us to conclude that the vaults were non-immunogenic. But what happens when we vaccinate a mouse with a vault that's packaged with a foreign antigen? I'm Lenny Rome, the Vault Guy. Tune into my next episode to find out if vaults, invisible to the immune system on their own, can be engineered to become powerful vaccine platforms. As always, please remember to like and subscribe and keep those questions coming.